So today I'll be speaking about engrams and its relation to the multiple memory system. So my key argument is that specific areas of the brain store different types of memories. And the importance of this topic is that research in this field could really help advance our understanding of memory disorders, such as dementia, Alzheimer's, and Huntington's disease, and could help potentially find cures and improve treatments in the future. So where memories are stored in the brain are known as engrams, and this is coined by Richard Semon, which is basically a physical substrate that helps in storing and recalling memories. And Richard Semon's definition of an engram is the enduring, though primarily latent, modification in an irritable substance produced by a stimulus. And what this basically means is that the exposure of a neural substrate to the stimulus that is being processed changes the neuron, and that is where the memory is then stored. So back to the key argument, this is in congruence with the multiple memory system, where separate memory systems are associated with activity in different brain regions. And this is all based on a study by Lashley and McCarthy, who had trained rats to navigate a maze without error. And after 10 days of that practice, the rats were able to do 10 errorless trials through the maze. And after the trials, Lashley and McCarthy had completely cauterized the cortex of the rats and repeated the training on the trials of the maze navigation after the cauterization. And what Lashley and McCarthy were able to find was that lesions towards the cortex had basically no effect on the rat's performance through the maze. And so the post-operative test results were not significantly any different than the preoperative test results. And what they concluded from this finding was that memory was not going to be localized to a specific area of the brain. So memory as general was found everywhere in the brain. So they failed to find an engram. So in contrast, the next study done by McCormick and his colleagues, they had potentially found a physical engram in the cerebellum for the memory of learned responses. And so how they did this was by recording electrophysiological activity during the classical conditioning of rabbits. And they recorded the neural activity in the ipsilateral cerebellum, which is one half of the cerebellum. And so they paired the conditional stimulus of tone, the unconditional stimulus of air puff with the conditioned response of blinking, and measured the neural activity and correlated that with the response. So after that, they had completed large ablations to the cerebellum and also other regions of the brain and repeated the conditioned response tests and measured the neural activity again of the blinking. And what they had found from this was that ablations to the ipsilateral cerebellum had completely abolished the conditioned response. So after completing surgical procedures on the cerebellum, there was no blinking response from the tone anymore. And this was cross-examined by creating ablations in other regions of the brain on other rabbits, such as the neocortex, where they had cauterized those areas, which had completely no effect on the conditioned response. And this was further again reinforced by, after the ablations in the ipsilateral cerebellum, they had trained the contralateral eye, which is the opposite eye with the opposite cerebellum on the other side of the brain, which was really easily able to pick up training on the training for the conditioned response of blinking to the tone. And what they suggested from this was that the memory of the learned response was retained in the cerebellum for quick relearning. And what this demonstrated was that memories of the conditioned response, at least to a degree, was localized to the cerebellum. And since learned responses are a type of implicit memory, that suggested that implicit memories were associated with the cerebellum as well. And this could be further affirmed by Pirani's study, Pirani et al. In 1993, and they affirmed the implicit memory storage of cerebellum alongside providing evidence for the multiple memory system, where different cerebral regions were associated with certain types of memory functions. And what they did through this was by measuring the memory abilities of 18 Alzheimer patients, 11 global amnesia patients, and 10 controls, and also measuring their hypometabolic rates in different areas of their brain as well, and correlating the two together. So the participants were based on the match pairs design. So all of the Alzheimer's patients and global amnesia patients were matched with controls that had similar educational and age backgrounds as the patients. And what their results suggest were that episodic and long-term memory impairments were found in both Alzheimer's and global amnesia patients, but there were only implicit short-term memory and semantic deficits in the Alzheimer patients. And this is shown on the image on the right side of memory deficit tests on the global amnesia patients. And for the metabolic test, they had measured the cerebral glucose to find the metabolic rates in these areas of the brain. So hypometabolism, which means low metabolic rates, were found in the hippocampus, the cingulate, and the frontal basal cortex for both Alzheimer's and global amnesia patients. However, for global amnesia patients, hypometabolism was only found in the thalamus. And for Alzheimer's, it was only in the frontal, parietal, cerebellum, and temporal associative cortexes. So what this suggests was that the deficits of memory were associated with the hypometabolism in these regions of the brain for each of the patient types of Alzheimer's or global amnesia. So what this suggests was that episodic memories, long-term memories, were associated with areas of the thalamus, the hippocampus, cingulate, and frontal basal cortex, whilst implicit memories, short-term memories, and semantic memories were associated with frontal, parietal, temporal associative cortex, and the cerebellum. And so on the bottom left of the slide, the image shows the distribution of memory systems, and the black dots or the black shapes, however, each black shape represents a type of memory, and they are found, and where they are on the brain shows which areas of the regions are associated with which type of memory function. So finally, I've discussed the strengths and weaknesses of all three studies. So with Lashley and McCarthy, there was a lack of demand characteristics due to the participants being animals, so they were not able to conform to any expectations that the researchers would have. And there was a reduced researcher bias, as they did not find what they were expected to look for, which means this counteracts any confirmation biases. However, there was low generalizability as there was an animal study. And for Pirani, they had used standardized procedure, which increases the reliability as all the trials use the same methodology. And the triangulation of methodology, which means they had used a wide range of methods, including the MRI, the PET scans, statistical analysis, and multivariate regressions, had increases their internal validity. And again, the match pairs design helps reduce fewer participant variables and reduce the chances of having a confounding variable. However, it again was a small sample size, and all the results were correlational, so no causation could be inferred. So finally, from McCormick, the researchers took an epistemological reflexivity and took that into account by using cross-examining with other brain regions, and also training the contralateral eye to increase their credibility of the results. And they also had used a microdrive system to operationalize the measurement of their neural activity, which increases their construct validity. But again, they had used rabbits as their participants, which decreases their generalizability. So to conclude, the evidence presented in the study do suggest there's a strong foundation towards brain regions being points of localization for storage towards certain types of memories. But of course, there is a need for further research in this field, and having advancing technologies in the modern day and age would really help give a broad range of research into this field and further expand this horizon. And each study presented in this presentation, as shown before, had added a new layer of understanding of where memory storage is in the brain. So the range of experiments and studies that could be done has been widened a lot in the future and in modern day due to the technology presented. Here are my references. Thank you.